Okay, the first question, when we deal with making the recombinant DNA molecules and transferring them to different types of hosts, we want to assure whether the gene that we have transformed, it has gone to the host genome or not, specifically in case of eukaryotic systems where we want to integrate the gene in the host. Okay, so there are two methods that we just discussed once. One of the method is that you isolate the total genomic DNA from your transgenic, putative transgenic. I use the word putative transgenic because before we confirm the integration, we can't say it as a true transgenic. Okay. Before confirmation, we always call that GMO as putative transgenic. When we perform two types of testings to detect the gene, presence of the gene, then we say it is a true transgenic. And those two techniques, they are, one is PCR, and we talked about PCR last time. We finished our lecture on that. And the second technique is using Southern hybridization. Okay. The PCR tells us about the amplification of that gene. If the gene is a transgene, it means it was not present originally in the host cell, but you have taken it from some other kind of organism and you have placed that gene in a different organism which does not carry naturally that gene. PCR is very efficient to detect that. Okay, you can PCR amplify the gene, you can clone it, you can sequence it and compare with the sequence of your construct, whether it 100% matches with your construct or not. Once you are done with that, you can say with confidence that this is no more a putative transgenic, it is a true transgenic, okay? Another method is the physical verification of the presence of the gene. Sometimes the PCR reaction can get contaminated with some other DNA which might have that gene of interest in that. And this happens very frequently actually. Okay? So PCR dependence is not good always. You have to confirm the presence of the gene by two ways. One is PCR and the second is Southern hybridization that gives you the physical presence of that gene. You can see the gene is there. Okay. Now, for this purpose, Southern hybridization is used, and the technique was established in 1975. Okay, and E.M. Southern, he was awarded with a Nobel Prize for this establishment of this. Now, I'm coming directly to the technique that how this works. This is mostly a technical lecture, okay? There's a theory behind it, but you studied that theory actually in the DNA modifying enzymes where we make the probes, okay? So on one side we make the probes, and on the other side what we do is we perform bloating. Southern hybridization is actually a combination of making probe and hybridizing the probe to your blot. You cannot say it as Southern blotting. Southern blotting is just a part of Southern hybridization. Is everybody clear? So what we do in this case, I'm sorry that we could not perform this experiment, but it is really very interesting to perform it. You have seen the electrophoresis gel apparatus. You have seen actually the lower part of the apparatus, which takes the buffer in it, and then there is a slab over on the top where you put your gel and you run the current so that your DNA can migrate along the gel. You use the same thing in Southern hybridization, but you don't use it for electrophoresis. You use it for a different purpose, Southern hybridization, And the setup is like this, that in the bottom, what you put is a blotting paper. And that blotting paper is immersed in a buffer, which is present in both of the chambers of the base of the electrophoresis apparatus. And this blotting paper runs throughout the top of that electrophoresis unit, and you place your gel, which you have already run separately. And this gel comprises of the total genomic DNA digested with a restriction enzyme that you used for actually cloning of your fragment of DNA in the genomic DNA. Okay? Or you can try any other restriction enzyme, doesn't matter. You can try more than one and run the different digested total genomic DNAs into different wells by digesting with different restriction enzymes. Okay? And then you put your gel here. Okay? And you put your gel upside down. Means the wells, they are facing towards the bottom of the electrophoresis system. On the top of this gel, you place a nitrocellulose membrane or a nylon membrane, which is known as high bond N membrane. Okay? That is positively charged membrane, and it is not fragile like nitrocellulose membrane. So what happens is the DNA is transferred from the gel onto the membrane if you put some more layers of blotting paper and a pile of tissue papers, and then a glass, and then a weight over it. So just by diffusion, the buffer enters into the first layer of the blotting paper, enters into the gel, and starts traveling to the top. Okay? So it's simply a diffusion of the buffer from the tanks to the tissue papers. And when the buffer moves, it takes along with it the DNA that has been resolved on the gel. Okay? So this DNA will migrate upwards, but it cannot pass through the nylon membrane because the porosity of the nylon membrane is so small, the DNA cannot pass through it. Because membrane is positively charged, the DNA will stick to it. And it will be an exact replica of your gel on the membrane. Is it clear?
Yes, sir.
Yes, sir.
Okay, so this process is known as blotting, that you transfer the DNA from the gel onto the membrane by a process of diffusion of buffer from lower part to the top. Okay, so this is called blotting, and it is a part of Southern hybridization. So now you can visualize that you need two things for Southern hybridization. One is a probe, which is prepared either by nick translation, random priming, 5' end labeling, or 3' end labeling. Okay, so once you transfer the DNA to the membrane, DNA is there, and you can use the membrane actually for hybridization with your probe. So this is a very simple explanation of the technique, very simple. But at this moment, I will make you realize that DNA is double-stranded when it is separated from the gel. Okay? And the double-stranded DNA cannot be used for hybridization. Hybridization always takes place between two single-stranded DNA molecules. Is it clear?
Yes, sir.
Okay, so it means that we need to denature the DNA when it is on the gel, because a denatured DNA can be transferred to the membrane. So it means that this gel requires very specific treatments. for the denaturation of DNA within the gel. Okay, so I will talk on this in detail as we move ahead. Okay, so the first concept that you get is you need a probe. You need a genomic DNA cut with different restriction enzyme and dissolved on an agarose gel. And the DNA is of double-stranded nature in the gel. So you need to convert it into single-stranded DNA by denaturation. You cannot do it with heat. There are other methods of DNA denaturation. There are three methods. One is heat denaturation. The second is alkaline denaturation. The third is acidic denaturation. Okay, these are the three things that you can do with the gel. Okay, so we use two of them to treat the gel, first with acid and then with alkali to denature the DNA. Okay, so I will elaborate that in the coming slides, but at the moment, I'm sure you are now familiar with two of the things that we are doing, two things separately. On one side, we are preparing the probe of the gene that we want to detect. And on the other hand side, we are taking the total genomic DNA from a transgenic, which is a putative transgenic, and we want to confirm the presence of that gene. And Southern hybridization is based on the formation of double-stranded DNA. When two complementary strands come together, they can bind to each other and make a double-stranded DNA again. And you remember that the probe that we prepare by either of the method, it is also double-stranded. So you need to denature the probe. Denaturation of probe is achieved by heating the probe to boiling point, like 100 degrees centigrade, for a few minutes. And the probe molecules are separated into single strands, and they carry radioactive nucleotides or fluorescent nucleotides. And if you put those denatured probe in a treatment of the membrane, then the single-stranded DNA of the probe can bind to the transferred DNA onto the membrane, which is single-stranded, and can do hybridization, resulting into double-stranded DNA formation, and you can detect the radioactivity where your probe has bound to the genomic DNA. Okay? So the key to this method is hybridization. It's a process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target DNA. If you are clear about this thing, then this is a Southern hybridization. That you have single-stranded DNA probe and you let it hybridize to a single-stranded genomic DNA, which is from transgenic organism. And if the hybridization occurs, the probe will stay on the membrane because it has bound to DNA. If the hybridization has not occurred during the washing steps, all the unbound probe will be removed. So the membrane will lose all the probe except at the positions where the probe has bound to the DNA, and probe stays there and gives radioactive signal or fluorescent signal at that particular position. Now, this is a calculation here. And this calculation is highly important because the first question when you start with a Southern hybridization by isolating the genomic DNA and running on the gel after restriction digestion, you have a question in your mind. How much DNA I should use? If you come up with small quantities of DNA, it cannot show you any results. If you go with a very high concentration of DNA, you cannot see the good migration of the DNA fragments. There will be an overloading effect. So you need to calculate how much would be the right concentration of the genomic DNA. How would you get an estimate for that? Now, for example, you know the genome size of the human beings. This is the genome size of a bacteria, 3 into 10 to the raised power 6. It's 3 millinucleotides big. What about the human genome? It's 3 into 10 to the raised power 9 nucleotides, which is much, much bigger. Okay, a bigger genome, you want to find a single gene, it's very difficult because you are having a lot of background of DNA which will not hybridize to your probe. A gene is a very small part of that. For example, if your gene is 3 kilobase long and you want to see it in a background which is 3 into 10 to the raised power 6 nucleotides, or in case of humans, if it is 3 into 10 to the raised power 9 nucleotides, how many copies of this 3 kb gene you need for detection purposes? Just like the copy number of the template DNA in PCR, where you calculate 1,000 copies. Okay, so there is a simple formula. You just think of using 5 microgram multiplied by, this is just a proxy, okay, that how many nanograms of the DNA of the gene will be there in the whole genome, okay. The minimum amount should be 10 nanogram, minimum amount, okay. For simplicity, what I have done here is I have taken 10 microgram as the total quantity of genomic DNA, which is going to be loaded in one well, okay. If we have 10 microgram of DNA digested with a restriction enzyme, and the total size of the genome is 3 into 10 to the raised power 6, okay? And the size of the gene is 3 kb. So this is the total quantity I am using. 3 kb is the size of the gene, and this is the whole genome size of the transgenic organism, okay? So what you do, you need to convert this 3 kb into 3,000 base pairs. So instead of writing 3 kb, you will write 3,000 divided by the whole genome size and the number of nucleotides, not in kb. Okay? It is in base pairs, means individual nucleotides. And now 3,000 are also the individual nucleotides. And 10 microgram is equal to 10,000 nanograms, because 1 microgram is equal to 1,000 nanograms. So 10 micrograms will have 10,000. So we have to bring everything on the same unit. Okay. So if you calculate this, the result of 3,000 divided by this 3 into 10 to the raised power 6, the value is 0.001. And when you multiply it with 10,000, it comes to be 10 nanogram. Now, if you do it with 5 microgram of DNA, then what will be the result here? It will be 5 nanogram. 5 nanogram is not enough for detection. Your maximum amount, the minimum amount should be 10 nanogram. So you need to adjust. So you can use this formula, 10 microgram into the genome size, but this is the variable. Your insert size will always vary, okay? Sometime it may be 1 kb, sometime it can be 2 kb, sometime it can be 3 kb. So you'll have to increase the amount of DNA to reach the 10 nanogram total transgene concentration. So what is this 10 nanogram? This 10 nanogram means your 3 kb fragment in 10 microgram DNA is representing 10 nanogram. Okay, so this is the basic formula. You can use this for the calculation that how much micrograms of the DNA you need for a particular size of DNA fragment that you are detecting, and that will give you the total quantity of your transgene in there. The minimum should be 10 nanogram. So you should modify this concentration to reach this value. Maybe if your gene is bigger, if this is 4 kb or 5 kb, you need lesser amount of DNA. Okay? But if your gene gets smaller, then this value can increase. So you always have to calculate this by just supposing a first concentration yourself. You can take it as 5 or 10 or 15, whatever you like. You just need to see that the value should be 10 nanogram at this position, at the answer level. Okay? When you get 10 nanogram at a specific DNA concentration, it means that your DNA is enough for going to Southern hybridization. You will be very easily be able to detect that particular gene transgene in the Southern hybridization experiments. There are 10 steps actually in Southern hybridization. First, I will describe only 5, which complete the whole Southern hybridization, but then later on I will expand those 5 into 10 steps. Just to give you a general idea, first what we need to do is a mixture of molecules is separated. What is that? That is your genomic DNA digested with a restriction enzyme and resolved on an agarose gel. Is it clear?
Yes, sir.
Yes, sir.
Okay, so this is the first point. The second is the molecules are immobilized on a matrix. You know the migration of DNA from the gel onto the membrane. That is high bond N membrane or nitrocellulose membrane. So you perform bloating. So this describes bloating. First, you separate the double-stranded DNA in the gel, then you perform bloating, and then the probe is added to the matrix. What is the matrix? Matrix is the membrane. You add the probe to the membrane for hybridization, okay? And then you remove the unbound probe by washing steps. And then what you do is you expose the membrane to X-ray film for detection purposes. This is a 5-step method that gives you that how you proceed for a Southern hybridization. Okay, so what I will do now is I will expand these 5 points into 10 points one by one, because it will be easier now for you to understand all these steps. That is stepwise procedure. Okay, and the first step is the DNA purification. You cannot do anything except you have purified the chromosomal DNA, the total genomic DNA, from transgenic organism, which you are calling putative transgenic. Okay? So there are several methods which are available. Actually, I'm not going for those methods. You will learn those when you get into a lab, that how to isolate the chromosomal DNA. But the step number 1 is DNA purification from the host cells. So after you have done the DNA purification, the second step is DNA fragmentation. And how you do that? It is simple, a restriction digestion. You digest that DNA in different tubes, 3, 4, or 5 different tubes, and a different restriction enzyme in every different tube. So you can try 4 or 5 different restriction enzymes. And you perform 4 or 5 restriction digestions. And after doing that digestions, you need to resolve that digested DNA onto a gel. Okay? And that is called gel electrophoresis. So first is DNA purification. second is fragmentation, third is gel electrophoresis, and you are moving step by step. So what you do, simply, you load the contents of the 4 or 5 different tubes, each tube in one valve. Okay? So if you use 4 different restriction enzymes, you will occupy 4 different valves. If you use 5 different restriction enzymes, you will occupy 5 different valves. Here you can use agarose or polyacrylamide. Polyacrylamide is actually for the very short DNA fragments. Most oftenly we go for agarose gel electrophoresis because the genomic DNA is much, much bigger in case of prokaryotes and eukaryotes. If you're using a virus, for example, then you might have to go to polyacrylamide gel because the viral DNA is very small. And when you cut it with restriction enzyme, it gives you very, very small DNA fragments which are not detectable by agarose gel electrophoresis. So you select polyacrylamide. Okay, so this is simply the gel electrophoresis on an agarose gel for the separation of the fragments of DNA that you fragmented by the restriction enzyme. The fourth is gel treatment. This is what I was talking. When the DNA is resolved on the gel, it is in the form of double-stranded DNA. Okay? In order to make it single-stranded so that your probe, single-stranded probe, can hybridize to a single-stranded DNA, you need to do some gel treatments before you proceed for blotting experiment. Okay? Blotting comes much later. You have to do it with the gel first, and that is called gel treatment. First treatment is acid treatment. It is known as depurination, SCL. When DNA is exposed to SCL, what happens? It depurinates the DNA. The purines are removed. And it's dependent on the strength of the acid, that how strong is HCl. If it is very, very strong, it will do a lot of depurination. If it is dilute, it will give you certain amount of depurinations, removal of the nucleotides. What will happen when there will be a removal of a nucleotide? क्या होगा फिर? डबल स्ट्रैंडेड डीएनए में से एक न्यूक्लियोटाइड आप निकाल दें तो क्या होगा? स्ट्रैंड ब्रेक होगा, है ना? Actually,
Yes, sir.
this is done very purposefully. अगर आपके पास 10 kb की fragment है and you want to the migration of this 10 kilobase big DNA fragment onto the membrane just by diffusion, it will be very difficult. The 10 kb will not move easily from the gel onto the membrane. So what you do is that you treat the gel with acid, and acid will give double-stranded DNA breaks, generating shorter fragments. That will be the same 10 kb, but it will be broken at different positions. And breaking the double-stranded DNA results into smaller fragment sizes at a particular position. And the migration of DNA will be facilitated on the smaller fragments. So depurination is mainly done to fragmentize, to break the double-stranded DNA backbone, so that the bigger DNA molecules, they could be converted to shorter DNA molecules. So what you do, you take the gel, you take it into a tray, and in that tray, you put 0.2 normal HCl solution. Just 10 to 15 minutes, leave the gel in that acid solution, and then take it out. The depurination has occurred. Then you take the gel to a denaturation solution. Denaturation means you want to separate the double-stranded DNA now in the gel into single strands. What you do? You put your gel in a 0.2 normal NaOH solution in another tray, and leave the gel for 10 to 15 minutes. All the double-stranded DNA gets separated into single strands within the gel. Okay, and then because alkaline pH is not good for nitrocellulose membrane and the nylon end membrane, so it is essential to neutralize the gel to the normal pH between 7 to 8, any pH like that. So you put the gel into a buffer, which is set at pH between 7 to 8, so that the alkalinity of the gel can be neutralized with this buffer. Okay, so gel treatment requires 3 steps. Number 1 is depurination, number 2 is denaturation, and number 3 is neutralization. Now your gel is ready for bloating. Okay, now you can go for bloating using the same setup that I was showing you in the beginning of this lecture. Okay, and that setup.
Sir, so why do we need smaller fragments of DNA for the blotting purpose?
For the blotting purpose, you know, the separation of the DNA fragments on the agarose gel is done with electric current. And the fragment moves according to the charge and size. When you are doing blotting, we are not applying any electric force, which will not push the DNA forcefully into the membrane. We are using the diffusion process. And diffusion, you know, it can migrate small molecules very easily as compared to large molecules. Okay, so that's the reason we are doing depurination, to break the larger fragments into sub-fragments, but we are not changing the migration on the gel. They stick at the same place.
Okay.
Okay, so the gel is treated 3 ways, starting from depurination, going to neutralization. And the next step is bloating. I have already told you through this picture that how we perform blotting experiment. This is the picture again. We have the gel tank, electrophoresis tank. We put the buffer in here. Okay, that is SSC buffer. You just need to remember the name, SSC. Okay, even you don't remember the name, you just need to remember it's a buffer. Okay. In the tanks here, we put the buffer, we put a gel, a blotting paper, which travels from one tank to the other tank, and it is sticking to the bottom of this gel tank. On the top of this, we place the inverted side of the gel so that the wells, they are facing the blotting paper, not to the top. Okay, this is very essential, highly essential. Okay, I will tell you the reason afterwards. And then on the top of that, you put the membrane and then the blotting papers and tissues to support the movement of this liquid towards the blotting paper size. Because it is dry here, it is wet here. The liquid will move through diffusion, and the smaller DNA fragments that we have generated by depurination and denaturation, they will now easily be capable of moving from the gel onto the membrane, and they will stick to the membrane. Okay, so this was the process. Okay, and this is another slide showing you the same, that the migration is towards the top. The liquid will enter from the tanks through this blotting paper, and it will flow directly to the tissue papers. But the DNA is shown here by blue colored arrows. This DNA will move from the gel onto the membrane and will stop here. It will just stick to the membrane. Because the membrane pore size is very small, DNA will not pass through. It will come out of the gel, it will stick to the membrane. Okay? So if you see the gel now under UV light, you should not be capable of seeing any DNA in there. No fluorescent DNA should be there if it is correctly migrated from the gel into the membrane. Okay? So the bloating is the purpose of moving the DNA from the gel onto the membrane. And the process requires diffusion of the buffer through the gel, through the membrane to the tissue papers, so that the flow of this liquid can help in the transfer of the DNA from the gel onto the membrane. Is it clear? After you have done blotting, what you do? You remove all these tissues. This pile of tissues is a simple tissue papers. Okay? And then you remove these two blotting papers. And then you remove the membrane and gel. You hold them both and pick them together. Don't separate them. Pick them together. The reason is when you pick this sandwich of the membrane and the gel and you place it on a glass plate, you will definitely go for the gel side up and the membrane side down. Now you have taken up both of these layers of membrane and the gel and you have placed it on a glass plate. Which side are the wells? If the gel comes to the top.
Upward.
Upward, yes, perfect. This is what is required. After picking this membrane plus gel together from this apparatus and keeping the gel along with the membrane on a glass plate, the wells are facing towards you. So what you do? You take a lead pencil and you dip that lead pencil into the well. Press it a little bit and you make an impression of the well onto the membrane using your lead pencil. Don't use a ballpoint. Don't use a marker. Don't use the ink. The washing steps, they remove all of those. We have having the impression of the wells by the lead pencil. The lead is not removed from the nitrocellulose membrane or high bond N membrane. It sticks and you will see that lead mark of the whole well. So that will tell you actually, if you lift the gel on the membrane, there will be no sign that where was actually the well side, which one is the lower side of the gel. So if you make the impression of those wells onto the membrane using your lead pencil, scraping down the well, the impression of the well is now transferred to the membrane. You will be able to see it just like you draw something on a paper using a lead pencil and it is visible. Now the position of the wells becomes visible on the membrane and you can mark the wells at number 1, 2, 3, 4. Tube number 1 was loaded here, tube 2 was loaded here, tube 3 was loaded here, and tube 4 was loaded here. So you need to keep trace that where did you load the first sample and in which position it should be on the membrane. Okay, so this is what you do. After lifting the membrane along with the agarose gel, you invert that, you put the marks of the well with the help of a lead pencil. Okay, that's what you need to do. And this is the completion of blotting. You have done the blotting. You have now the membranes with the image of the gel on it, just representing the well placement. nothing else. You will not be able to see the DNA. Don't touch your membrane with the ungloved hands. Okay? Always wear gloves when you are performing bloating. Okay, the DNA from your broken cells can also stick with the membrane and can give you actually a background fluorescence or background hybridization when you perform the experiment. So how many steps we have gone? We have gone 5 steps, and there are 5 steps left. So we need to go one more lecture to complete this Southern hybridization.